Greetings from NASA headquarters in Washington, D.C. I'm Dr. O'Neill, and I'm here to help walk you through the steps to take until I'm able to meet you out in Alaska. I would just like to let you know that NASA takes any threats of life extremely seriously, and I urge you to use the utmost precautions since we do not know what we are up against yet. First, we must discover if the unknown life contains the four biomolecules: carbs, lipids, proteins, and nucleic acids. When testing for sugars, heat a solution and add iodine to the mixture, and it will change from a blue to a red-orange color. Complex carbohydrates can be detected by adding starch to iodine, and the solution will turn from a dark purple-brown color. Next, test for lipids by adding Sudan-3 and mixing it. The solution will make two layers if lipids are present: a base layer, which would be yellow, and a top one, which would be a pale pink or orange. Then, test for proteins by mixing it with a Biuret solution, which will change from light blue to pink or purple if proteins are present. Test for nucleic acids by adding diphenhydramine and placing it in a hot water bath. The solution will turn purple if DNA is present and green if RNA is present. If it is not present, it will remain clear. If it does have the four biomolecules, then we must decipher if it is a prokaryote or a eukaryote. Prokaryotes are simple cells without a distinct membrane-bound nucleus or special organelles. While eukaryotes are an organism whose cell contains a nucleus within a membrane, they also contain complex organelles such as a nucleus, endoplasmic reticulum, cell membrane, Golgi, ribosome, and chloroplast. You can tell the difference by the fact that eukaryotes are relatively large, around 30 to 150 nanometers, while prokaryotic cells are relatively small, around 0.5 to 10 nanometers. They also have a membrane-bound structure, while prokaryotes do not. There are 7 organelles found in a eukaryote: a nucleus, mitochondria, endoplasmic reticulum, Golgi, cell membrane, ribosome, and chloroplast. You can identify a nucleus by getting a sample of the unknown life and using a stain called hemotoxylin. It will stain the nucleus a blue, violet, or brown. The nucleus's main function is to contain a majority of the cell's genetic material. Mitochondria are identifiable under a microscope by using a stain called fuchsin. They will also have zigzag crescent formations found on them. The mitochondria has a major role in synthesizing, folding, modifying, and transporting proteins. It also moves proteins to the Golgi. The Golgi is identifiable under a microscope by using the dye N-actate galactomic saminal transferase 2 to identify the Golgi with our fluorescent fusion proteins. They will look like a dark semicircular black rings near the bottom of the culture. They are responsible for transporting, modifying, and packaging proteins and lipids into vesicles for delivery to targeted destinations. The cell membrane is identifiable by staining it using a light cell reagents for plasma membrane by Thermo Fisher. They keep toxic substances outside the cell. They have receptors and channels to mediate cell activities between organelles and the cell. An outside environment, separating metabolic process within organelles. Ribosomes are cell structures that make proteins. They are identifiable by using a co-massy blue staining from Thermo Fisher Scientific. Chloroplasts are identifiable under a microscope by using a white stain. They also reside in plant cells as oval bodies with white dots inside of them. They are food producers in plant cells. They convert light energy of the sun into sugars used by the cells. If they don't have all four biomolecules present, then we next must test if it can be a virus. Viruses are non-cellular microscopic parasites that replicate only inside of living cells, the other organisms that depend on cells for metabolism and reproduction. They are exceptionally small, and their size range varies from 20 to 200 nanometers. Viruses are parasites and cannot thrive without a host. They depend on cells for metabolism and reproduction. They have no cytoplasm or organelles. Take a tissue sample of the potential virus and expose it to a range of potential hosts and test the results. They are not considered true life because they are non-cellular and only show some of the 8 characteristics of living things. Next, we must test if it interacts with the environment by testing if it has a metabolism and responds to stimuli and adapts. Most life, as we know, use respirations to take up and release substances. In addition, a majority of life uses glucose. So perform a 3-part test, first by giving an organism a fixed amount of glucose and observe what happens to it. Then repeat with a fixed amount of oxygen. Lastly, observe the amount of carbon dioxide produced by the organisms. If it takes in glucose and oxygen and releases carbon dioxide, it has a metabolism and reacts to the environment. In order to test if it responds to stimuli and adapts, set up a thermotest by taking an example of the cell and put it near a heat source and observe if it moves closer or farther away from the heat. Repeat, but use a cold source and observe the changes in adaptation that the organism makes. Viruses do not have a metabolism because they use their host as an energy source and release replications of themselves to infect their host further. They do not respond to stimuli and adapt in the way that humans do, by touch, light, or heat. But they do respond to their host dying by procreating copies of themselves to further infect their host. Prokaryotes and eukaryotes do not respond to stimuli and have the ability to move what they stimuli. They respond to light, oxygen, and magnets. They do have an aerobic and anaerobic metabolism, while some are phototrophs, which derive their energy from the sun. Another important step is testing the unknown life's ability to reproduce. You can test this by taking an example of culture on the side and examining it underneath a microscope. Record how many cells you start off with and wait 24 hours. Reobserve the cell and record how many there are now. For example, if you started out with 5 and you now have 10, mitosis or meiosis must have happened and the cells must be reproducing. Viruses' main purpose is to reproduce and inject nucleic acid into a cell, and then produce a virus coat and nucleic acid. Most prokaryotes and eukaryotes reproduce asexually. Sexual reproduction in eukaryotes results in a genetic offspring, while prokaryotes result in a clone of themselves. We must next ask ourselves if the unknown life is complex or composed of many different cells. Viruses are single-celled organisms that are not complex, but prokaryotes and eukaryotes are complex. They contain many complex traits that humans have and obtain a similar chemistry. Cell growth can be measured by first recording the cell's basic parameters, mass, volume, and the ratio of the two. Use a suspended microchannel resonator with a culture counter to measure mass, volume, and density during the cell process. If the density, mass, or volume of the cell increases, then the cell has grown. The cell membrane controls homeostasis by maintaining their stable internal environments when dealing with external changes. For example, controlling the volume of the cell and the water movement entering and leaving the cell. Positively and negatively charged molecules cannot pass the cell membrane without transportation. Living organisms detect a change in their environment. Change in the environment conditions outside the cell from very cold to very hot and see if the shape changes. If the shape changes, then the function has changed. Under a microscope, you can view a cell taking in water, and then when it would have too much, it would push them out. Now I must warn you, if what you discovered is a virus, bacteria, eukaryote, or prokaryote, you have found life. The first thing you must do is immediately go into a level 4 quarantine and lock down the site of the unknown life. Nobody goes in or out with my permission and must be wearing protective suits and hoods. I've contacted and alerted the Surgeon General of what has happened, and he has ordered an official quarantine. We will set up a contamination tent that has been airlocked and equipped with an air pressure changing system. This will be able to trap airborne pathogens. This will have a double door system so that scientists never expose unknown life to the air. Anyone who has come into contact with the unknown life without proper safety gear needs to go immediately isolated and go through extensive testing and 24-hour examination. From now on, anyone providing tests to the unknown life must be properly equipped and undergo 7 minutes of being sprayed by water and virus chemicals over the hoods and the suits before the doors will open. They're not allowed to leave the facility until repeating the proper decontamination process. I urge you to use the utmost precautions, and I will be there soon.